Hey biology. Today we are going to take some notes on evolution. We're going to start off where you stopped the other day in your fill in the blank notes, and we're going to get through the rest of these introduction to evolution notes, evidence of evolution, and how we see this actually happening today so that we are ready to go, ready to jump into some other stuff, taxonomy and classification and all of those kinds of things here on Tuesday when I get back. Okay, so that is what we're going to do. Make sure that you have your notes out on your desk, ready to go. And we're going to start right where it says the fossil record. So you should have put in lots of notes here in this area back on Thursday. So like you should have drawn in some kind of bones here. That was like the worst bone ever. My pen is not cooperating with me very well today, so hopefully it will when we are actually writing words in here. But I drew like a fish head right here and some bones. There we go. I'm getting the hang of it now. All right. But over here, this is a bone. Oh, there. I have to label it. Okay. And we said that this was the youngest layer. The top layer is always the youngest and the bottom layer is always the oldest. So that's something that you need to remember, okay? So whatever you draw in here, okay, you need to have something in each one of these layers so that you can say, hey, let's see, this dinosaur head down here, okay, this teeth, all right, and an eye. Okay, this is older than this malfunction looking bone up here, which is younger. Make sense? All right. So let's make sure that you have everything filled in the blanks. Some classes got some things filled in and some classes didn't. So I'm going to go quickly through this. If you need to, pause at the end and I will go slower on the rest of this. All right. So fossils are the preserved remains or traces of dead organisms. Okay, so this can be anything. This can be bones that we find right here, like we drew in, or it can be footprints. It can be poop. Okay, yes, we have fossilized feces from prehistoric organisms. It can be any kind of trace. All right, they're usually found in rock, but sometimes they're found in permafrost, which is permanently frozen ground, or encased in amber. Okay, permafrost can be found in Antarctica or in Greenland, the North Slope of Alaska. Amber, you're probably remembering Jurassic Park. This is fossilized tree sap. We call all of the fossils found the fossil record. Okay, it's important to know the age of each fossil so that we can work out how species evolved over time. Okay, so right now in these layers, the only thing that we know is younger and older. We don't have an exact date. We just know that the bottom layer is older and the top layer is younger. So that's our age right now. Scientists can date fossils using the layer of the rock they're found in. The oldest rocks are always found in the deepest layers. That's what we're showing over here. This is known as the law of superposition. Underline that or box it or something like that. The law of superposition. So that just means that younger layers are at the top, older layers are at the bottom. They can also directly date fossils using radioactive decay of certain elements, such as carbon. So we can take a little bit piece of this fossil right here and take that out and find how much carbon is in it. Knowing the radioactive decay rate of carbon, we can tell how old it is. And there you go. So that's your part for the fossil record. Pause here if you need to make changes to your notes. Otherwise, we are moving on. So, next part, how does the fossil record provide evidence of evolution? And this is the whole point right here, right? Evidence of evolution. How? So, fossils show us evidence of evolution through the gradual change of physical structures over time. Okay? So for example, we have fossils of early horses, that's what we're looking at right down here, that had multiple toes. And we can see how this evolved over 50 million years to have a single toe or hoof, okay? So it started out with multiple toes. That's what we're looking at right here. 50 million years ago, we had multiple toes. 1, 2, 3, 4, okay? And then as we get younger, we have one that only has 1, 2, 3 toes. We get a little bit younger. We get a longer leg and a longer middle toe. 1, 2, 3. And then eventually those other 2 toes are gone. We don't need those anymore, and we just have 1 in the middle. And then in present day, this is our horse, and it has a large hoof to support that large leg and the large body. Okay, so I want you to note these are oldest and these are youngest. All right. So now, fossils of organisms that are in between 2 species are called transitional forms, okay? So Archaeopteryx, there you go, that is the correct pronunciation of this right here. Archaeopteryx fossil is a transitional form between a prehistoric reptile and a bird. So we can see this because we've got claws up here, claws. And then we have feathers right here. The claws are more reptile-like, and the feathers, so birds don't necessarily have claws like this, and the feathers are more bird-like. The reptiles, true reptiles, don't necessarily have feathers like this. So that's why this is noted as transitional. There are many examples, but we don't have fossils of transitional forms for all living species. Okay, so that's something to note. A lot of times nowadays you will hear of people saying that dinosaurs were birds. Okay, not necessarily, but they have bird-like features, and this Archaeopteryx right here is what we think is a transitional fossil between a true reptile, which we used to think dinosaurs were true reptiles, and true birds. So it's possible that there are more transitional species in between there that could give us some more evidence for that transition. All right, we're going to keep on going. Next, So big question. Why are there gaps in the fossil record? Okay. We talked about this little bit on last Thursday when I was showing you my geologic timescale up there above the board, right up there above. Okay. So I said that we don't have records of every single living thing that lived. Instead, it takes certain conditions for an organism to fossilize. So this is what we're going to write down about that. Most organisms that have lived on Earth did not leave any fossils behind. So that's what we're saying. Most organisms did not leave fossils behind. Why is that? Okay, so the conditions for fossilization are very rare. Okay. Number 2, some organisms are soft-bodied and do not have hard parts to form fossils. So this is one of the key conditions that you have to have. You have to have hard parts like a skeleton. So you have to have bones, or you have to have a shell, or you have to have teeth, or you have to have some kind of hard surface in order for there to be a fossil. Soft-bodied organisms that don't have any of these kinds of things right here are very, very, very rare to see a trace of. And so that's why we think there are so many more things that used to live on this earth that we don't even know about. They just were never preserved. Geological activity has destroyed fossils. So volcanoes, earthquakes, any kind of tectonic activity where you have tectonic plates moving against one another that have crushed rocks and things falling on each other, that's all going to destroy these really, really delicate traces. And then it is unlikely that we have found all existing fossils. So that's when we come over here to this picture, okay? Here's a paleontologist who's looking for fossils, and they're digging in one specific spot, but they don't find anything. Ooh, sad. I've been on many expeditions like that. Don't find anything at all. Guess there aren't any fossils here. Ooh, well, I'll try somewhere else. Well, guess what? There's lots of fossils. They just didn't dig in the right place. Okay, that's what all we're saying. So it's important to remember we don't have a complete fossil record, but we are looking at and we are making scientific guesses based on what we do see. All right, let's continue. You're going to go to your next page for notes. All right, this is page number 2 for your evolution notes. And we're going to start right up here where it says embryonic development. Here's some more evidence for evolution. Okay. We're going to start with the fill in the blanks, and then we'll come over and do some doodling. Embryos are the early stages of development of a living organism. By studying embryonic development, we can see the order in which different structures and organs emerge. Okay, so that's what we're looking at over here. We're looking at embryos, so the very early stages of an organism that has just been fertilized, and their cells are dividing, and their nerves are telling certain cells to make certain organs and all sorts of things, and this is what development is taking place, and this is what it looks like. Now, early embryos of different vertebrate species, what does vertebrate mean again? Well, it means that it has a backbone. okay? So any organism that has a backbone is a vertebrate. We are vertebrates. Fish are vertebrates. All sorts of things that walk around are vertebrates because they have to support legs and their entire body. But the early embryos of different vertebrate species are hard to distinguish or tell apart. They have homologous structures. Ooh. What does that word mean again? Homologous means similar. Okay? Remember from our first page of notes, homologous structures, including a tail. Ooh, we talked about that before too. Eye spots and gill slits. These are not evident in the mature forms of all species. Okay, that is important to note. Their presence in early embryos is evidence that they evolved from a common ancestor. All right, so we're looking at the embryos or the early stages of development of multiple different species. Okay, so again, we're going to say, hey, this line right here, this arrow, it is development. Okay, from the early stages well, I'm having trouble with my pen here. Okay, early stages of development to late stages of development. All right. Now, this first one right here, going straight down, is a fish embryo. All right, starts out looking like this. The second one is a turtle. Turtle embryo starts out looking like this and going into the late stages of development. And the last one is a pig embryo. Okay, starting out, ooh, we're going to draw it, and then going down like that. So notice that at the very early stages, the fish and the turtle look quite similar. Okay, so they both have these features right here. This is the eye spot. Okay, they both have gill slits. And they both have a tail. All right, so what should the pig look like? Well, probably pretty similar. Okay, so you're gonna see mine, and you know that I don't draw wonderfully, especially with this kind of a pen here on the computer, so feel free to laugh, but I'm gonna try my best. Okay, here is that nose looking thing, and then we need some gill slits, and then we're gonna have a tail. I made my tail like really big. It's totally up to you, but you at least have to have an eye spot. Okay, there's my eye spot, there's my gill slits, and then maybe they have this little thing right here, I don't know. And then there's the tail, eye, gill slits, and tail. Who knows? Maybe you can draw a little bit better than me. All right, so as the fish develops, then it retains the eye, retains those gill slits, and retains the tail, but it all straightens out, more streamlined. And then late embryonic development looks very much like a fish. The turtle retains that eye. It still has some gill slits here in our middle embryonic development, but they're gonna get lost here by the later stage. And then here we still have a tail, but look, we're starting to develop a arm or a flipper right here and a feet right there. All right. So in the late stage, you can see the eye is still there, more keenly developed. We've got a mouth instead of those gill slits, and we've got legs with flippers attached, and it would still have that tail right there. And we've got the starting of a shell. With the pig, our two embryos at the middle stage look very similar to each other. They have the legs developing right here. They retain that eye and retain those gill slits, and then we still have the tail. But look how different they are here in the late stage. All right, eye, mouth, and the legs right here instead of flippers, and no shell. All right, so that's what we want to be looking for here, because we have this right here. This is our evidence. All right, this is what we're taking a look at as evidence of a common ancestor. All right, pause if you need to, but I'm going to show you a little bit more on embryonic development. You do not have these slides in your notes. I'm just going to take a look for a second. So using this picture, can you tell which one is the human? All right, we've got embryos from 1, 2, 3, 4, 5 different organisms. Okay, developmental stages of 5 vertebrates. That's what we see down here. Remember, they all have backbones. So can you tell which one is the human and which ones will not be human? Okay, place your bets, because I'm gonna go and reveal here in a second. I'll give you another clue here. How about now? Can you tell which one is the human? Which one's not gonna be human? Okay, well, I probably would guess this one is not going to be human, right? We do not have a tail, we don't have flippers, and our body does not look like that. What about this one, this one, this one, this one? What do you think? I'm not sure. Okay, all right, let's keep going. Ooh, now can you tell which one is the human? Hopefully, yes. Okay, so these are the later stages of embryonic development. But do you see right at the beginning, they all look pretty similar. All right, at the end, not the same at all. Let's see what they are. We've got a fish, a turtle, a bird, a pig, and a human. So there you go, just driving that point home. All right. So now let's go down to molecular biology. All right. When we talk about molecular biology, I want you to think DNA. Okay? DNA. That's all we're talking about here. We've got this huge strand of DNA right here, and don't think, oh, this is hard stuff. No, we're just talking about DNA. All right? So first, let's come over to our DNA strand and label as much of this as possible. So this right here, this that makes up the side of the ladder of this big double helix, all of that. This has one strand right here and one on the other side, okay? This is our sugar, phosphate. What's the word I told you to remember? Backbone. Okay, sugar, phosphate, backbone. So remember, DNA deoxyribonucleic acid. Okay, so that's your sugar phosphate backbone made of deoxyribose and phosphate. All right, now what about the other things? All of these right here, this, all of those. So those are bases. Okay, specifically nitrogen bases. Okay, so remember A, T, C, G, that's what those are. All right, so now that we've refreshed our memory on all of that, let's go over and fill in some blanks. Every living organism contains DNA, which is the heritable genetic code? Genetic code of life. All DNA is constructed of the same 4 nitrogenous bases. That's what we're talking about right here. The same 4, okay? Held together by a sugar phosphate backbone. Boom. Right there. The presence of DNA in all living organisms suggests we all evolved from a single common ancestor. Okay. Another homology. Homology. Remember homologous structures? Homology just means similarity. Another homology or similarity is found in the synthesis of proteins. Remember protein synthesis? That's just making proteins. So if you want to say making, right up here. The process requires RNA to be produced from DNA in all organisms, from turtles to bacteria. And the same amino acids are coded for by the same DNA triplets. Okay. So let's look at the bottom right here. We can sequence the genome or genetic material of different species and compare to see how closely related they are. So we're comparing other species to the monkey right here by shading any differences in bases in another color. All right, so we've got an animal. We've got a monkey, and this is its short DNA sequence. This is just a gene right here from a monkey. We've got CATCGATT. All right, a hawk and its gene, squirrel, its gene, and a frog and its gene. So what we do is we take that monkey, this is our comparison right here, and we're going to say, okay, does everybody have a C in the first one? Yes, everybody does. Does everybody have an A in the second one? No. That one is different. And then the frog also has a different one, right? Okay, so let's keep going. T. Everybody has a T. Everybody has a G or a C. Ooh. G, we've got differences right here. That was a C right there. A, everybody's got an A. T, everybody's got a T. T, ooh, this one, everybody is different there. Okay, so now we can see which ones are different and how they're different. So the next question says, which species is the monkey's closest relation? How would we figure that out? Well, we take a look at the hawk and we see it has 1, 2, 3 differences from the monkey. The squirrel has 1, 2 differences from the monkey. And the frog has 1, 2, 3 differences from the monkey. So, which one has the fewest differences? My squirrel. Okay? The one that is most similar or has the fewest differences is the closest relation. So, the squirrel. Does this make sense? Okay, hopefully it does. You're going to, if you need to pause here. Otherwise, we are moving on to biogeography. Okay, biogeography. This is one of my favorite subjects to talk about because I get to talk about plate tectonics and all the shifting of everything way back in prehistoric times. So, here we go. What is biogeography? How does it provide evidence of evolution? So, biogeography is the study of the distribution of organisms on the earth. So, how are organisms distributed in different areas on continents and in oceans and all over the place? Okay, are there certain organisms that are concentrated in one area? Are they not? And when did they get to that area? How did they get to that area? That's what we're talking about with biogeography. So patterns of distribution are best explained by evolution. And I'll explain how here in just a moment. For example, species with common features can be found in similar environments. Where they would have experienced similar selective pressures. Okay, so let's continue with our filling in the blanks, and then I'll come over here to this diagram. Our knowledge of the movement of tectonic plates further helps us to understand the distribution of organisms. So if you don't know what tectonic plates are, it's just the crust of the earth, basically, that has been broken up into multiple different pieces that are kind of like puzzle pieces, and they can move toward each other, or they can move away from each other, or they can scrape side by side each other, and they've been moving like this throughout the history of Earth, ever since it was formed. So the continents that we know today, the distribution of where they are today, is not the way it's always been. In fact, we actually had a supercontinent back hundreds of millions of years ago called Pangaea. Okay, there are other times when we had multiple different supercontinents like Laurasia and Gondwana. So we're going to take a look at that more specifically. For example, broad groupings of organisms that existed before the supercontinent Pangaea split up can be found all over the world today. Those that evolved after the split into Laurasia and Gondwana are only found in certain parts of the world. The presence of unique species like koalas, found only on isolated islands, is evidence of evolution. Okay, so let me give you an idea of how this works. In Pangaea, there we have a huge supercontinent. We've got all of these different parts, like this part right here. This is what would eventually turn into North America. Okay, this down here is South America. This over here, this whole mass right over here, this is Africa. This is up here, this is Europe and Asia. Okay, this is India. This is Antarctica. And this is Australia. All right, so you got kind of an idea of what's going on there. Let's see. Come on. There we go. I'm gonna see if I can change my ink. There we go. All right, so let's say we had a organism that lived in Africa, and it lived not only in Africa but also in India, Australia, and Antarctica. So this whole entire blue area right here. Okay? So that was its distribution. You've got an organism that it's able to travel between all of this because all of these continents are locked together. They're right next to one another. There's no ocean in the middle there. And this is, you see within these lines right here, this is a specific temperate latitude that allows them to live anywhere along those lines. Okay? Now, we go down here. And it starts to break up. So here is Antarctica that has a little piece of that organism. India is going to have quite a lot of that organism. Australia hasn't broken quite off of Antarctica yet, but they're still going to have a little bit right here. And then you've got some that are in Africa also. Okay? So now they're separated, all right? They all millions of years ago were the same organism, but then they became isolated, all right? So we can see fossil evidence of them, but then once they became isolated, then they started to evolve to the area that they were in. This one became different than this one, became different than this one, became different than this one. All right? And that's what we see evidence of. So an organism like the koala here on Australia is so separated from everything else, it's isolated, so you don't see anything like a koala anywhere else in the world. All right? That's what we're talking about. Okay, so the last little bit here, the last part is down here at the very bottom. Okay, so let's see if I can go back to red. Okay, so let's fill in some blanks. All right, direct observation. So this is something that when evolution happens and we can see it. We can see it. Okay? Now, this doesn't happen with fossils or with transitional species or biogeography. We can't see that happening. All right. Direct observation is something that where evolution is happening so fast, we can actually see it. Okay. So evolution occurs consistently. Okay. Consistently and constantly. Let's put the word constantly in here so that we understand it happens all the time instead of consistently. Let's do that. So all the time. Normally, it takes a very long time to see the impact of the physical structures of a species. However, sometimes we can witness evolution in action in just years, months, or even weeks. Okay? This can happen due to strong selective pressure. And when we're talking about selective pressure, we're talking about natural selection. And we're going to talk about that here this week, okay? And a high rate of reproduction. So you got to have a lot of babies for this to happen. Okay? So one example with this would be bacteria. All right, antibiotic resistance is a great example of direct observation of evolution. So that's what we're taking a look at here. All right. So we have bacteria that has become resistant to methicillin. It's an antibiotic. You know, when you get an ear infection or something like that, the doctor prescribes amoxicillin, okay? Or sometimes maybe you've had penicillin, okay? That is an antibiotic, all right? So we've got a whole bunch of different bacteria in here. The regular bacteria look like this. They're just regular bacteria. They've got a cell membrane and they've got this linear DNA in the middle of them, okay? But then you've got an irregular bacteria, okay? And this one is resistant. He's like, haha, guess what? I'm gonna take over. No, I don't know. Okay, maybe he has something different about him, but that's his resistance. All right. So, if that happens, then they're going to put the methicillin into this petri dish, and which one's going to survive? This one. Your weird one. Okay, the one who's like, yeah, I survived. Okay, nobody else is left but him. Okay, now how do bacteria reproduce? Do y'all remember? They just split in two. Do they need a partner to reproduce? No. Okay, so this guy right here is free to do whatever he wants. He's survived through the methicillin attack, and now he's going to reproduce, and there's going to be a lot of him all over the place. All over. Whoop. Okay? All he has to do is split in two. Okay? And because there aren't any other bacteria in there, the regular bacteria, they're all gone. They got killed by the methicillin, okay? Because there aren't any other bacteria in there, we say that means there's no competition. So, which one's going to take over? The one that is resistant. Okay? Is methicillin going to work on this group of bacteria? No way. All right? So, let's describe this in words. Number one, a mutation occurs that gives a bacterium methicillin resistance. Number 2, when methicillin is used, what's happening? Right here. All other bacteria are killed. Okay? So then, what happens here? The methicillin resistant, that's because that's what they are, bacteria reproduces rapidly. Okay? The methicillin-resistant bacteria reproduces rapidly. So that's something that we can actually see. So when your antibiotic stops working, that's probably because you have resistant bacteria in there and they have to find a different medication in order to get it. All right. So there you go. This little bubble up here is not necessary, but this right here is another example of resistant populations. You can write whatever you like in here. All right. Eek. All right. So that's it. That's all of your notes here for today. You need to make sure that your name is up at the top of this page, Evolution Notes number 2, and turn it into your sub, please. Thank you so much, and I will see you all tomorrow. Bye.